So as in the last video we have discussed about the structure of the kidney. In today's video we are going to discuss the structure of the nephron which are present in the kidney. Alright, so in each kidney we do have 10 lakh nephrons. Alright, we do have two kidneys, so in one kidney we do have 10 lakh nephrons, in another kidney we do have 10 lakhs nephron as well. Alright, so total we have 20 lakhs nephron in both kidneys. So now we are going to understand the structure of the nephron, how it looks like. Alright, so first of all we do have glomerulus. Initially I am going to make the Bowman's capsule. Alright, so here I am going to make, so this is Bowman's capsule which is shaped like a cup. This is what? This is Bowman's capsule. So I should write this as well. This is Bowman's capsule. So here I am going to make arteriole, which is afferent arteriole. Alright, this is afferent arteriole, and we know that the exchangement or the filtration happens from the capillaries, not from the arteries or the veins, because of their sizes. Alright, so it will get divided into the capillaries. These are capillaries. Alright, and then it will get out from the efferent arteriole. So this is what? This is glomerulus. So let me write here glomerulus. And this is what? This is afferent arteriole and this is efferent arteriole. Alright, so now I am going to make here the PCT. So I should make this with color. So here I have PCT which I am making right now. This is folded. Alright, this is the tube which is folded. So here I have PCT which is proximal convoluted tubule, which is known as PCT. Alright, this is PCT which is known as proximal convoluted tubule. I should write this with white color so that it can be visible. So let me write this again with the white color. Alright, so proximal convoluted tubule, and the abbreviation of proximal convoluted tubule is PCT. Alright, so this is proximal convoluted tubule. Now we are going to make loop of Henle. Alright, that should I make with this color. So here I have a long branch of this tube which is going down. Alright, and again then it is going up. Here it is. So this is what? This is loop of Henle. So I should write this with white color. Loop of Henle. This is descending loop of Henle, this side, because it is like coming from the upper and going down. So this is known as descending loop of Henle. Descending loop of Henle. while this side is going from the down to the up. So this is ascending. Alright, so this is ascending loop of Henle. Ascending loop of Henle. So as you can see here, we have made descending loop of Henle and ascending loop of Henle. Now we are going to make distal convoluted tubule. Alright, so that I am making with the same color. Alright, so here I have distillated convoluted tubule, distal convoluted tubule. So I should write this distal convoluted tubule. The abbreviation of distal convoluted tubule is DCT. Alright, so here we do have distal convoluted tubule. Then it is entering to a tube which is known as collecting duct. Alright, so I am making here the collecting duct. So here is the collecting duct. I should write here with white color. This is what? This is collecting duct. Alright, so this is the structure of nephron we do have. First of all, we do have glomerulus from where the urea is coming. Alright, then it will get filtered. Alright, here the filtration will be happening, and that filtration would be ultrafiltration, which means filtration would be done under pressure. So that would be called as ultrafiltration. Alright, so here what things would be coming? Urea would be coming. I'm not going to write here, but I'm going to speak. Alright, so urea plus H2O plus ions would be coming here to the Bowman's capsule. Alright, so this is basically, I do have filtrate. Alright, so this would go same in the PCT, and from the PCT reabsorption will happen of the glucose, of the water, and that would be active reabsorption. Alright, then it would be going to the loop of Henle, initially in the descending loop of Henle. Then it will maintain the concentration of the ions, because one of them is permeable to the water and another is permeable to the ions. So they will maintain the osmotic pressure or osmotic regulation, and this filtrate will go to this distal convoluted tubule, and then when this will enter into the collecting duct, it will known as urine. Alright, so this would be then known as urine. So this is the structure of nephron we do have. So I should write here the structure of nephron. Yes. Structure of nephron. Alright, so here I have the structure of nephron. Now let me tell you one more thing. Like this, from the efferent arteriole, now some more capillaries will be formed, and those capillaries will be known as paratubular capillaries. So I am making this here. Alright, one will go like here too. Again it will come here. Alright, so these are known as what? These are known as paratubular capillaries. So I should write it down, and the capillary which will go to the loop of Henle will be known as vasa recta. Vasa recta. and ultimately these all capillaries will combine here together and they'll form vein. All right, so I am writing here, this is known as renal vein, or renal vein. All right, so this is vein I do have, and those were paratubular capillaries. All right, so now here is a important thing we should know, that what is that? That is glomerulus plus renal tubule, which means this glomerulus and plus these all renal tubules, because proximal convoluted tubule is a tubule, then we have loop of Henle, that is also tubule, distal convoluted is also a tubule, so that is whole known as renal tubule. All right, so we do have two parts in the nephron. First one is glomerulus, which is comprised of capillaries. All right, then we have tubule, all right, renal tubule, which includes Bowman's capsule, PCT, LH, which means loop of Henle, and DCT, which means a distal convoluted tubule. Now, here is an important thing, a very interesting thing, we should know that as well. So, here glomerulus, like as you can see here, this glomerulus plus this Bowman's capsule combinedly known as Malpighian body. So I should write this down, Malpighian body. All right, so here I am going to write here Malpighian body, which is the combination of what? Which is the combination of two things. All right, what those two things? Let me write here with the white color. All right, so that is the combination of glomerulus plus Bowman's capsule. So Bowman's capsule and glomerulus combinedly called Malpighian body. Bowman's capsule. Alright, so as you can see here, Malpighian body, which is the combination of glomerulus and Bowman's capsule. Alright, so here we need to know that we do have two types of nephrons. Alright, first we do have that is cortical nephron, and second we have juxtamedullary nephron. Now we should talk about their weight. Alright, so initially I'm going to write here types of nephron with this color. Alright, so I am going to write here types of nephron. As I told you, we do have two types of nephron. Two types of nephron we do have. So I'm going to make the arrows first and second. Alright, so let me write it down. First we do have cortical nephron. Alright, and second we do have juxtamedullary nephron. Juxtamedullary nephron. Alright, two types of nephrons, juxtamedullary nephron and cortical nephron. Now we should talk about their weight, like how much they are present. Alright, so let me write here down. So cortical nephrons are 85%, which are not like helping in anything. We do have so many cortical nephrons, but we do have only 15% of juxtamedullary nephron. And it is also said that the one who are changing the society are in less number, and the one who are doing nothing, they are in more number. So cortical nephron are doing nothing, but they are in 85 number, like they are doing normal filtration. All right, and juxtamedullary nephron like it controls the concentration of urine. Either it should be concentrated or diluted, so that is controlled by juxtamedullary nephron. So juxtamedullary nephron are only 15% in our body. Alright, so now they have gotten these two names like cortical nephron and juxtamedullary nephron. I hope you guys are getting something. Like cortical is coming from the word cortex. Alright, so cortical nephrons are present where they are present in the cortex of the kidney. As I told you, we do have two parts in the kidney. First one is cortex, the outer part, and the inner part is known as medulla. Alright, so nephrons which are present in the cortex of the kidney are known as cortical nephron, and the nephron which are going to the medulla are known as juxtamedullary nephron. Alright, which controls the concentration of the urine. Like either it should be concentrated or diluted. That is controlled by juxtamedullary nephron. So I should make here the kidney so that it can be more clear to you. And I should tell you guys the location of these nephrons. So here I am going to make with this color. Alright, so here our kidney is this very okay. So I am going to make here again. This is the kidney I do have. Here what we are talking about the location of the nephron. So location of the nephrons, like where the cortical nephron is present and where the juxtamedullary nephron is present. As I taught you, we have two parts of kidneys. So the outer part is known as cortex. This was the cortex. We have discussed these all terms in the last video. Alright, so this was cortex and this was medulla, in which medullary pyramids was present. Medulla, because we know that medulla was subdivided into conical surfaces which were known as medullary pyramids. So I should make here those. These were medullary pyramids, and in each kidney we do have range of 16 to 20 pyramids. All right, so these were known as medullary pyramids because they were present in the medulla. So medullary pyramids. Alright, so these are medullary pyramids. Then they were opening in the thin tubes which were known as calyces, minor calyces. So here these are. Here these are. Here I am making the minor calyces. Then it will combine together and ultimately form the major calyces. Alright, so here the ureter was coming out from the body, not from the body but from the kidney. So this was the ureter. These were the minor calyces, calyx, and this was the major calyx. Alright, and the part of the cortex which was entering to the medulla was known as renal column or column of Bertini. So here I am writing renal column, or it is also known as column of Bertini. Alright, so now here we are talking about the location of the nephron, like where cortical nephrons are present and where juxtamedullary nephrons are present. Alright, so here I am going to make, so I should make this with this color. Alright, so cortical nephron, as I told you, these are present where? These are present in the cortex of the kidney. So here they are present, loop of Henle. Here these are present. These are known as what? These are cortical nephron. I should write here cortical nephron. And what's the percentage of cortical nephron we do have? That is 85%. Alright, so now I am going to make here the juxtamedullary nephron. So as you can see here, this is the PC. Just difference is here what the loop of Henle is going to the medulla. As you can see here, I am going to make again loop of Henle is going to the medulla. So that's why only just because of the loop of Henle going to the medulla is making it juxtamedullary nephron. Juxtamedullary nephron. Juxtamedullary nephron, and their percentage was 15%. Alright, so this was 15%. So now, as in the today's video, we have talked about the structure of the nephron and types of nephron. In next video, we would be going with the functions of the nephron. So this was all for today.